This time I'm going to focus on the past 10,000 years. And you may be thinking, what's so special about the past 10,000 years? Well, what is special is that before the last 10,000 years, give or take 1,000 or a couple thousand, we were living as hunters and gatherers of our food and our clothing and our homes. And we were using mostly stone tools. And so this refers to the Paleolithic. The Neolithic is the term that we use to refer to what happened with the advent of agriculture. So in the past 10 to 12,000 years, we started to domesticate plants and animals. And this map up here, this global map, notice, illustrates some of the areas, most of the areas in the world where major crops were domesticated. Now, the earliest domesticated animals and plants come from the Middle East. You've heard of the Fertile Crescent in Turkey and Iraq and Iran. And those are, in fact, where the earliest examples of, say, wheat, barley, about 10,000 years ago, or rather 10,000 BC, are found. But there also were areas of domestication throughout the world. There were areas of domestication in East Asia, in sub-Saharan Africa, in North America, in South America. So by no means was this something that occurred just in one place. But it is the case that domestic plants and animals were domesticated in the last about 10 to 12,000 BC, since 10 to 12,000 BC. So you just saw a map of where the initial points of domestication occurred. This is a current map from the UN Environmental Protection Division that illustrates areas of the present-day globe that are covered by agricultural land or grazing land. And you can see that there is a lot of it. Lots of the globe is covered, is either agricultural or grazing land. So that tells you that in 10,000 years, this has been really successful. These isolated areas of domestication have expanded into all of these areas of the use of land for domesticated plants and animals. There were many consequences of this. The landscape changes as a result. The settlement pattern changes as a result. The intensity, the types of social interactions change. Our diet, our nutrition, our diseases all change. So the environment has been very, very different over the past 10,000 years than it was previously. And we might expect that human evolution might have occurred then also with this major shift in environment. We also created the first settlements that we would call, first of all, settlement, that we actually settled down instead of lived in mobile camps, let's say for 3 or 4 months at a time, and then moved again after 3 or 4 months, which is what we did while we were living as hunters and gatherers. We now see year-round settlements. For example, one of the first towns is reconstructed here. It was a town called Catalhuyuk in Turkey. They were mud brick homes. They dated to about 6,500 years before the Christian era. Soon thereafter, we had small urban areas. We moved up from town to urban area. And these all of this was taking place, occurring in this Fertile Crescent area. Small town or small urban area, as long ago as 3,800 years before the Christian era, and a little bit larger town a little bit more recently. So now you're starting to think about, okay, people are settling down, they're living close to one another, they're living in the same place year-round. This also seems to have been very successful. Here's a map from 1970 to 2000. This was looking at major urban areas throughout the world and the increase in the extent of urban land area over just this 40-year or so, 30-year period. And my point is that, wow, if you go back and you think, oh, we're starting to think about the first towns 6,500 years before the Christian era, and now we're talking about, increasing about 58,000 square kilometers of agricultural land in only 30 years, and major metropolitan areas all over the world. Agriculture, domesticated plants and animals, again, very successful. So yes, agriculture does have its advantages. It can create surpluses of food. So like these Andean highlanders, in front of a big mound of potatoes, you can store food. And hunters and gatherers are known for a number of reasons, one of which is they don't store food. They collect it, eat it, collect it, eat it. So they only have as many possessions as they can carry from one camp to the next. But if you're settled and you're an agriculturalist, you can grow more food than you can eat in a day or two, and you can store it. And this is a reconstruction of one of the earliest food storage, grain storage structures, again in the Middle East, in the Fertile Crescent area. You can simply support more people. And I don't even need a map to show you that human population has been increasing and exploding in the rate of increase recently. We're now over, we're over 6 billion? Are we over 7 yet? 7. We're over 7 billion. Agriculture also has some disadvantages. It can lead to pollution. You can imagine, what do you expect? Even with a small town of people living close to one another, you're going to have sanitation problems. You're going to have garbage problems. It can lead to environmental degradation. It can lead to conflict over land. Now there's such a thing as good land, bad land. It can lead to a decline in biodiversity, both in the environment and in the diet. So here's an illustration of what a field looks like after preparation for slash and burn type agriculture. They've been burning the forest here in Indonesia. So what about health and nutrition? A little surprisingly, health and nutrition were poorer after the introduction and the transition to agriculture. There are a couple of dozen places in the world where we have enough skeletal evidence, the remains from burials and cemeteries, that we can actually look at the health of the population while it was still living the Paleolithic lifestyle of hunters and gatherers, during the transition to agriculture, and during the early years of agriculture. So we can look, to the extent that we can tell from skeletons what health was like, we can look at what happened during that transition, and in most cases, not all, but in most cases, agriculture and animal domestication was associated with poorer health status and nutritional status. Usually, we get shorter by 2 to 5 inches. Now, that's a substantial decrease in adult height. Usually, we see an increase in the number in teeth with cavities. This is taken from some work done by Clark Larson, who's from the Ohio State anthropology department, who's very well known for his studies of the effects of the transition to agriculture. And this is a site that he worked on where there were several time periods, prehistoric time periods, while people were hunters and gatherers. And you can see that fewer than 5% of the teeth had cavities. Then agriculture was introduced. Whoa! And here we are up to 25% of the teeth having cavities. It's the same population, just before and after the transition. We also see evidence of increasing wear on the teeth, more lost teeth post-transition. Other indicators of nutrition are bony indicators of anemia. And for example, this is a sign, cribra orbitalis, of anemia. Porotic hyperostosis, a sign of anemia. We start seeing more signs of damage due to infection on long bones. Now, the domestication of plants and animals dramatically changed our diet. My plate has replaced the food pyramid, right? That we're a little bit more used to, and that my plate tells us that we should eat a balance of fruits and vegetables and cereals and protein and some dairy products daily. Well, and this is based on sound scientific principles. It supposedly also is based on the latest in nutrition and an evolutionary perspective. Well, let's take a look at that. What did we eat before we were agriculturalists? Well, we can tell that by studying modern-day hunters and gatherers, people who don't have any fields or any domesticated animals. And we see that they rarely ate grains, if at all. They would just, maybe sometimes of the year, they might collect some wild barley, some wild wheat. They got more protein than we do now. A bigger percent of their calories are from protein. And they got more fruits and vegetables than is recommended by the food pyramid, or I mean the My Plate. You can see I'm a food pyramid trained person. And there are no dairy products. So the transition to agriculture was a big change for us, for our diets. And we have adapted genetically to some, although apparently not all of these dietary changes. We do have some changes in our gene pool that reflect the high starch diet that we now have, with lots of grains, more refined sugars. We have adapted to drinking milk after weaning. We've adapted to be able to sense more toxic chemicals in foods. Here's one example. The amylase protein is called AMY. It's the protein, it's an enzyme that digests carbohydrates. It gets its name because another word for starch is amylum. And individuals here in this room probably, we vary in the number of copies of the amylase gene. So not only can genes be present in different variants, they can also be present in different copy numbers. And the more copies of the AMY1 gene an individual has, the more amylase he or she makes, and we can find in the saliva. So this illustrates that. Here are the number of copies of the amylase gene. They range from about 2 to 16, and the larger the number of copies, the greater the concentration of amylase in someone's saliva. Okay, so, well, let's look at some populations and see if Populations that are eating a lot of starch, cereals, for example, a lot of rice, a lot of potatoes, a lot of corn. That has only been possible since the transition to agriculture and the domestication of rice, potatoes, and corn. And we see that populations eating the high starch diet have a median, that is 50% of the population has more than 6 copies of the salivary amylase gene, 6 or 7. Populations eating the older low starch diet, high complex carbohydrates, such as modern day hunters and gatherers, tend to have a median around 4. So the difference is not huge, but the difference is in the direction of selection increased the copy numbers of the amylase 1 gene, and we are more efficient at digesting amylase. We have also adapted to drinking milk after weaning. And this is something that you probably know a little bit about because there's more written about it. And there's something called lactase persistence. If you're an adult and you enjoy sitting down to a big glass of cold milk, chances are good you are lactase persistent. And in order to do that, you have inherited 1 or 2 copies of an allele for continuing to make the enzyme lactase throughout your life. If you do not care to sit down to a tall glass of milk, like me, then chances are you are recessive for that particular trait, and you stopped making lactase around the age of probably around 4 or 5. So what does the lactase enzyme do? In milk, there is a unique protein called lactose. I'm sorry, it is not a protein, it is a sugar, lactose. The lactase is the protein. And the protein cleaves lactose into glucose and galactose that can be absorbed into the system. So in order to get the calories from lactose, from drinking milk, you have to have lactase. If you can drink milk, or you can eat dairy products rather, such as yogurt, and the bacteria have already done that breakdown. So you can be lactose you can be recessive for that. That is, you can be lactase intolerant and still eat dairy products because you don't have the problem of having to break down the lactose yourself. And the frequency of lactase persistence varies globally. We see a high prevalence in Northern Europe. We see a high prevalence in the US, where we're descended from Northern Europeans largely. We see a high prevalence in Australia, again, because we're descended from Northern Europeans. We see a low prevalence throughout China, throughout the New World, in sub-Saharan Africa. So basically, it's mostly Europeans and the rest of their descendants in the rest of the world who make a lot, who have a lot of people that are able to digest the milk and lactose, and a few from sub-Saharan and East Africa. What do we know about the use of milk? Well, the earliest evidence that we have of people using milk in its liquid form is from about 6500 BC. So we domesticated sheep and cattle and so forth thousands of years earlier. But apparently we were using sour milk, yogurt, and cheese, and not milk until fairly recently. So why would there be an advantage to drinking milk in its liquid form? Now, you can get the calories, you can get the nutrients and so forth from the cheese and the yogurt. So what's the benefit? Well, first of all, Sugar, lactose, does provide calories. There's a protein in milk called lactoferrin that is an antibacterial. So it gives you a little bit more protection against bacteria. Milk is a very high percentage of water. So here's a clean source of water in a time when sanitation was becoming a problem. And milk may also enhance the absorption of other nutrients. So there may be a lot of reasons for selection for the ability to digest the lactose in milk and to be able to drink milk in its liquid form. Now, interestingly, if we look at the genes for lactose tolerance, they are different if we look at Europeans and their descendants, and if we look at East Africans. And this quote comes from a science article. There's more than one way to have your milk and drink it too. And among Europeans, there is a single mutation in a gene that controls the production of lactase, this enzyme. And it's the one that accounts for the lifelong ability to digest lactose. That allele is absent in Africans. Instead, there was a different mutation in a gene that controls the production of lactase. So here we've got different mutations occurring in different parts of the world, responding differently in the sense of increasing in allele frequency in response to what we hypothesize were the same selective factors. And this is an example of convergent evolution. Two or more populations independently evolved the same trait. So we've got two different populations, the Northern Europeans and the Sub-Saharan Africans, under similar dietary pressures. And they converged on the same solution. And we can also estimate how long it took. So it took probably 6,000 to 8,000 years in Europe for almost everyone in Northern Europe to be able to digest lactose, and probably even a shorter time period in East Africa.
I'm curious about 2 points that you made. First, going back to that cluster of mud homes from 6500 BC. When you see agricultural areas in Europe, the homes, you can look through the community and see the fields in the back. Here you have a cluster of homes that implies separation of skills because they're not immediately adjacent to the fields. And the second question is that with this milk, there was a slide that you had that in going from a hunting way of life to an agricultural, people became shorter in stature and had different problems associated with it. So one can infer that it wasn't necessarily a positive way for their bodies to switch, that the bodies are reacting. I know, and all this milk, people are warned today with osteoporosis not to drink milk.
I didn't know that. So if you have osteoporosis, the recommendation is don't drink milk?
Right, that it tears away from the calcium in your body.
Interesting, I didn't know that. Okay, to answer your first question, it was a small village, so it was still near the fields. It's not like they had to walk miles to get to the field at the early agricultural sites and the early towns. So, however, you are correct that one of the things that occurs, one of the social changes that occurs as we develop the ability to accumulate food and so forth, is that then we do start seeing specialization. We see, some people maybe as we develop, for example, we learned how to make pots. We invented pottery. So there would be someone making pots and maybe trading them for food. So that does go along with differentiation of kind of economic specialization. With respect to the changes in health, you raise an interesting question, and that is, why would people do it? And there are a couple of hypotheses. Why would we do something that might not be better for our health? One, maybe maybe there was population pressure. I mean, you do produce more food. And so if there was population pressure on the land, then if you can make your own food and think that you're in control of your own food production, that would be a powerful stimulus. Right? So there was ecological change around that time. Maybe people saw, I'm speculating here because there are a number of hypotheses. The environmental change was things became warmer, climate became warmer, it became easier to grow things. So it's not that people suddenly woke up one morning and said, I will be a farmer. People had been practicing or doing little forays into to gardening, if you will, for thousands of years before that.
After the beginning of agriculture and domestication of animals, excuse me, there was a large increase in population. Isn't it possible that the reason we're seeing negative effects is because these people would have died? They wouldn't have survived prior to agriculture.
Oh, you mean they might have been less, you're thinking that perhaps it's not because of change in diet that there was more illness, but that because of the change in diet, more ill people survived. Let me think about that. Well, we should be able to, you got me a little bit stumped, so I'm hypothesizing here. And one thing that I would suggest doing to address that is to look throughout the life cycle. For example, if your hypothesis that it's simply sicker people who are surviving, we should see evidence of sick people in children, early in childhood, in both non-agriculture and agriculture, and then we should see it not in adulthood in the non-agriculture, so that then the evidence that we have that people were sick would be associated with an early age at death. And I don't know the answer to that. So let's move on to tasting bitter substances. With the new environment, with the new nutritional environment, inventing foods, domesticating foods, we had a lot of new foods to try. And not all of them were necessarily good for us. Some of them may have had high levels of cyanides or other things that were not particularly good for us. So the fact that many of us, although by no means all of us, can taste bitter foods is hypothesized to be related to the fact that many toxic foods are toxic because they're producing chemicals to prevent themselves from being eaten by herbivores, like us, or other animals. So, how many of you, anyone here who does not like broccoli? We all like broccoli. Oh my gosh. Someone will admit to it.
With or without butter and salt?
No butter, no salt.
Oh, they're not.
Yeah, okay. No butter, no salt. Okay, so we've got a couple who don't care for broccoli. For those, how about people who do not care for grapefruit? Nice sour grapefruit, more. Okay. Well, those are two foods that contain plenty of bitter toxins. Coffee, how about, do you like your coffee black? No milk, no sugar. Okay, some people, maybe that's because anyone not like their coffee black. It has to have milk and sugar. A few of you, okay. So if you like milk and sugar in your coffee, and if you don't care for broccoli that much, you don't care for kale and cabbage and grapefruit, you may be a bitter taster. That can be formally tested. I was thinking about bringing the tests with me tonight, and then I thought it was not nice to do to the tasters, because you have to taste something terrible if you are a taster. So there's a question then. Does the ability to taste these bitter substances relate to what you actually eat during, under normal circumstances? And does that in turn influence your health? And so we can ask, does it influence our food preferences, alcohol intake? We can also use this knowledge that some people are tasters and non-tasters to figure out the chemistry of foods, the chemistry of taste, and alter foods so that we like the way they taste. So for example, we could perhaps grow a new broccoli that everyone loves, and then everyone will eat it and they'll get their antioxidants. Are we the only animals that can taste bitter substances? First of all, I should say, how do we even know there is such a thing as the ability to taste bitter substances? Well, the reason we know that is during the 1930s in a lab, a chemistry lab, someone was synthesizing something called phenylthiocarbamide, also now known as PTC, and dropped some. And the dust was in the air in the lab, and some people said, ugh, God, that stuff tastes terrible. And other people in the lab said, what are you talking about? And so they became puzzled, and they worked out the fact that this particular chemical could be tasted by some people, who we now call tasters, and could not be tasted by other people, whom we now call non-tasters. This was in the 1930s, and it was very interesting, and people started running, scientists started running around and seeing, if Lithuanians can taste, if Parisians can taste, if Zimbabweans can taste, and so forth. And then someone said, gee, I wonder if other animals can taste. Let's test the tastes, of non-human primates. And so then they said, well, how are we going to do that? How are we going, the typical way to test, to do the testing is to prepare a half dozen small glasses of water with different concentrations of this bitter chemical, PTC. And so then, the person takes a drink of one and says, don't taste anything, 2, 3, and so forth. And eventually we see how strong does the solution have to be for an individual to taste it, if at all. So the thought was, well, can we do this? Could we do this with a chimp? Could we give it a little array of glasses and ask it to please taste cup number 1 and so forth? And so one of these guys said, oh, I'm going to give it a shot. And so he took his testing kit and he went down to the Edinburgh Zoo, where they had a chimpanzee, female chimp, well known to the public, used to the public. And he handed her a cup of the strong PTC, the bitter water. She took a sip and she threw it in his face. So I'm anthropomorphizing a little bit, but I think that she may have been a taster. We now know that in fact chimps do taste, and they do have the same gene. They have a mutation in the same gene, but it's a different mutation than we have. So we have evolved these bitter taste sensitivities separately, chimps and us, since we diverged. So we talked last time about the proximate cause and the ultimate cause. What's the proximate cause, kind of the biochemistry and the anatomy? It's taste buds. And this is I thought about asking you guys to do this too. And I figured you'd probably decline. But you can paint your tongue with blue food coloring, and you can look at your taste buds, and you can actually count them. We have about 6,000 of them covering the tongue. And tastants, such as broccoli or such as fine wine or chocolate mousse, interact with the pores on the taste buds, as is illustrated here. And then there are nerves that go from the taste buds to the brain. So that's how we taste things, whether it's sweet or salty or bitter or sour. And bitter taste is coded for by a whole family of genes called the TAS2R genes. And it's influenced by another factor. So I already mentioned the non-taster and the taster phenotypes. That is, can you taste it or not? That's your phenotype. And they correspond to known genotypes at one of these loci, the TAS2R38 locus. You don't need to worry about the names, of course. Then there's a second factor, and we don't know what causes variation in the second factor yet. And if you have more of these particular type of taste buds called fungiform papillae, then you can be what is called a super taster. So there are three possible tasting phenotypes: non-taster, taster, and super taster. And I have a picture up here of my friend Linda Bartoshuk. Linda Bartoshuk is a psychologist who is the person who unraveled all of this about super tasting and has pursued the psychophysics and the behavior associated with taste. And she was the one who identified super tasters. So one of the things that this illustrates is that there's a very complex relationship between our genes, between other things that happen during our lifetime. So it's not only what your individual genotype, but it's your own life experience that influence what your phenotype is. So let's imagine that we've got genotypes for non-tasting. We'll call it AA. And then we've got genotypes for tasting bitter substances. We'll call them AP or PP. And we've got, so that's our genotype for the taste 2R gene, 2R38. Do you have a lot of fungiform papillae or a few? You could have few, you could have few or many, you could have few or many. I'm being very qualitative here to make my point. Then, so if you have the non-taster phenotype or genotype and few fungiform papillae, you're going to be a non-taster. If you have the genotype for tasting and you have few or you have many fungiform papillae, you could be a taster or a non-taster, and part of that will depend on whether you had ear infections during childhood. Oh, what? Ear infections influence my taste? Yes, because the nerve that goes from your taste bud to your brain passes near your ear, and it can get damaged if you have a lot of severe ear infections during childhood. She was telling me about when she was first working out the genetics of tasting and non-tasting, and she kept getting all of these kids whose parents didn't match the kid's phenotype and inferred genotype. She thought, is there a lot of illegitimacy here, or is something else going on? And the university shall remain nameless. It's not ours. And then she figured out that there was this other factor, this life experience, that was influencing whether or not an individual actually had a taster or super taster phenotype. So you have to have the right genotype, you have to have lots of papillae, you have to have no childhood ear infection in order to be a super taster. So it's starting to illustrate how complex it is for natural selection for us to figure out how natural selection works. Now, just in case you're curious, how many super tasters are there likely to be? In the general population, say if we were to test ourselves here, probably about half of us would be tasters and a quarter would be super tasters. That's what Linda Bartoshuk found. Now there is geographic variation. For example, nearly 100% of Native Americans are either tasters or super tasters. So that 25% of, it's 75% among other populations, yet 100%. One hypothesis about, gee, why do Native Americans have such high levels of tasters, and that relates to the risk of iodine deficiency in getting goiters. There are some foods that have chemicals in them, bitter chemicals, that are also goitrogens. That is, they bind iodine, so that even if you have iodine in your diet, you don't have much to absorb. So the hypothesis there is that, oh, if you're a super taster, you might eat slightly less, of that food and be less likely to develop goiter. Now, this, I usually have my students test themselves. So Anthropology 103 is Introduction to Human Evolution. And it's been a while since I had them do it because I ran out of papers. But I'm getting more from Linda, so there'll be another set. In the fall of 2006, I had 29% tasters and 43% to super tasters. Wow. So I had a lot more super tasters than might be expected. I actually had one student in one class, I think it was, who said, "That was the worst thing I've ever tasted in my life. I couldn't do that again if I had to." He had tasted this, it's a little paper impregnated with the chemical, and he made a beeline out of the room. I give everybody candy, but candy wasn't enough. He needed water. So there is variation, among samples of undergraduate students here at Case Western. The biological variation can have influence on dietary behavior. Perhaps this explains partly why some children are picky eaters. They're very sensitive. People who are tasters, or in particular super tasters, tend to eat fewer fruits and vegetables, so they're getting fewer antioxidants, have a slightly higher risk of some forms of cancer. At the same time, people who are super tasters also are super tasters of the creaminess and the oiliness in fat. So they don't like dessert. Yeah, right. Or they eat fruit for dessert. Well, they don't like fruits either. What do they eat? I don't know. Non-fat things. So they tend to be more slender. Not, skinny, but more slender. They eat fewer goitrogens. They drink less, have less alcohol intake. So there's subtle changes in behavior that are all related to risk factors for modern day diseases. So this is a really interesting area to continue to look at. Now also, since the Neolithic, we have occupied a huge range of new environments and encountered new selective factors. So I'm going to talk about high altitude natives, because while there were occasional people living at altitudes above 12, 14,000 feet before the Neolithic, basically permanent habitation at high altitudes in the Andes and in the Central Asia and in East Africa took place in the last 10,000 years. So what we're going to ask is, well, do highlanders from these various areas differ from their likely ancestors? Is there evidence that selection may have acted to improve the ability, to live under that stress? So, what's the stress? The main stress at high altitude is something called high altitude hypoxia. So we've got, on this map, we've got an illustration of altitude in meters. So 4,000 meters corresponds to about a little over 13,000 feet. 4,400 meters is about the altitude of Pikes Peak. And the barometric pressure decreases. Oxygen is about 20% of the, contributes about 20% of the barometric pressure at all altitudes. And this scale on the right tells us the percent of sea level in terms of oxygen that we're inhaling. So by about 13,000 feet, every lungful of air has only about 60% of the molecules of oxygen as here at sea level. Now this, on the one hand, is a wonderful stress because you can't avoid it. So all ages and sexes and health statuses and rich and poor and so forth are exposed to this stress. It also is a very severe stress because we have to have oxygen. You can't go on an oxygen diet. You have to have a continuous supply. So how might we have adapted? Well, this is something called the oxygen cascade. It describes the transport of oxygen from the air to our lungs, to our hearts, to the bloodstream, to our muscles, and to the mitochondria where it's burned to make the energy for us to survive. And this shows us the amount of oxygen here at sea level at those different places, and the amount of oxygen at high altitude in Peru. And look at this, at every single step of the oxygen cascade, there is very little oxygen. So, what are we doing? Well, we are descended, this is not new. Although going to high altitude is new, and being exposed to that form is new, being exposed to hypoxia in general, in fact, has a very old evolutionary basis. Our ancestors 2.5 billion years ago, cyanobacteria, evolved in water that had a very low oxygen tension. Multicellular organisms evolved about 2.2 billion years ago, and again, in water in very low oxygen tension. Furthermore, even after more animals and organisms evolved, and we were living on land, well, this is before we were living on land, but look at this. In the past 600 million years, there's been huge variation in the amount of oxygen in the air. We all learn in school, 21% of the air is oxygen molecules. Well, nowadays it is, but there have been times when it was as high as almost 30 and as low as almost 12. Now we're descended from all those organisms. Not all of them. I mean, our ancestors are, we can make a tracing back here. So we are descended from ancestors who have lived through millions of years of hypoxia, as well as hyperoxia. Furthermore, all of us experience hypoxia in everyday life. In utero, we were all hypoxic. You've heard the phrase, Everest in utero. Yeah. And many developmental processes rely on being appropriately hypoxic. We become hypoxic while we're sleeping. Our wounds are hypoxic. This is included here. It's not a healthy blood smear. It's an anemic blood smear. But anywhere between a quarter and a half of the globe has iron deficiency anemia. So we're used to adapting to that. And in addition, then, there are various diseases like COPD, like congestive heart failure that are associated with disease and everyday life. So we and other multicellular animals have a gene-based oxygen homeostasis system. We have genes that are oxygen sensors. We have genes that are transcription factors that tell other genes, whoa, we're hypoxic, turn on. And then we've got these target genes. These are the ones that turn on. And I'm going to talk about HIFs, hypoxia-inducible factors. These are the transcription factors. Target genes that you've heard about include EPO, as in, I was going to put a picture of Lance Armstrong, and I just couldn't bring myself to do it. It's too sad.
In your lifetime, you can be a super taster or a non-taster, depending on your age. I mean, children, normally they won't eat an onion on a sandwich, for instance. But as you get older, you tend to accept those things. And even older, not that you can eat just about anything, as well as environmentally. I mean, we here are adapted to our own food sources, but if we go like to Tibet, would I be able to eat some of that food? I don't know, but yet I can drink black coffee.
Yeah, actually, yes, you'd be fine with barley. But I don't know about tea made with butter and salt. You're absolutely right. And if I were going to treat this more completely and just have a whole lecture on this, it is definitely the case. And you mentioned two things that we'd have to take into account. One is, what do you get used to eating, right? And that is, if you grow up in a culture where you routinely eat bitter food, this might be normal, and you eat it. So that would be another thing that we would need to investigate, is the extent to which the interaction there between a particular cultural preference and tasting. The second thing that we would want to take into account is just what you said, changes in the number and sensitivity of taste buds across the life cycle. And we know that in, like, say, the decades of the 70s and 80s, people lose their sensitivity, and their sensitivity changes. And actually, you know, for example, my mother complains that she can't taste anything. And I've asked Linda, I said, okay, what can we do for Mom? And she says, nothing. Yes, you're quite right. When we go to altitude, what happens? Our hemoglobin concentration increases. We increase our ability to transport oxygen with our hemoglobin. Andean highlanders do the same thing. Here's what they do. They go to high altitude, or they live at high altitude. The higher the altitude, the higher the hemoglobin concentration. What about Tibetans? Oh, look at this. No. They go higher, but their hemoglobin doesn't increase. Well, that's a puzzle. I wonder why. Maybe they're all sick. Maybe they all have iron deficiency anemia. No, I looked at that. So what we see is that the acclimatized phenotype is not shared by Tibetan highlanders. This is looking at Tibetans, this is looking at Han Chinese. Maybe we shouldn't, and another set of acclimatized individuals, and the Han Chinese have more hemoglobin than Tibetans. So for a long time we said maybe there's a genetic difference, but we didn't know what the genetic difference might be. But perhaps now, with an improvement in genomics, we can test that hypothesis. See, here's what, we already had that. Genomic analyses support this hypothesis that Tibetans have a distinctive gene pool. And in fact, they have a high frequency of particular alleles in the oxygen sensor genes, a high frequency in one of the hypoxia-inducible factors, called EPAS1, and a high frequency at certain target genes. All of these are in the direction of association with low hemoglobin concentration. And here's an illustration. Nowadays, we don't have to just look at the taster gene or the lactase gene. We can look at throughout the genome. And this was an example where we looked at 600,000 different loci on chromosomes 1, 2, 3, 4, 5, and 6, and so forth. And we looked at the allele frequencies. It says stop.
No.
Skip malaria.
Do this, don't do.
Okay.
People want to know about it.
Okay, well, I have to talk. I will speed up a little bit. And so what we did is we said, what's the allele frequency for Han Tibetans? Han Tibetans are the ancestors of Tibetan, of modern. Did I say Han Tibetans? Yes. Wrong. Han Chinese. Han Chinese ancestors are the ancestors of both modern-day Han Chinese and modern-day Tibetans. And they went to altitude, and so we can look at modern-day Han Chinese as representatives of the ancestors of modern-day Tibetans, and we can say, well, what nucleotide do they have at a particular locus? What's the frequency for Tibetans? What's the frequency for Han Chinese? I just made this up. It's close to actual values, and then we can do a chi-square test, and we can say, oh, there are different allele frequencies. The T allele is present in much greater frequency among the Tibetans. So we do this 600,000 times. Now, if you know anything about statistics, you know that chances are good you're going to get some statistically significant results if you do 600,000 tests. So we have to correct for that, and this red line represents that correction. If there is a p-value above this, that means that even corrected for having made 600,000 tests, we have a high allele frequency among Tibetans. And look where that was. 600,000 genes we looked at, and the one with the allele frequency different that was statistically significant was a hypoxia-inducible factor. Hallelujah. Well, did it have anything to do with hemoglobin concentration? Well, let's see. So we have this hypothesis. Okay, here's the ancestral gene pool where we've got little a and big A, let's just say. And 90% of the genes are little a. And now it's 400 generations later, 10, 11,000 years. And now in the descendant Tibetan population, it's the opposite. We've got 90% big A genes. And we know how it's inherited and how it associates with hemoglobin concentration. So we are able to actually look at, okay, you've got parents who are heterozygous, you've got children who have a possible range of genotypes. The more Tibetan alleles you have, the lower your hemoglobin. So this tells us that natural selection has been selecting to have lower hemoglobin at altitude. Now, why would that be? And in fact, we now have evidence from a lot of different samples. If you're a homozygote for the, quote, Tibetan alleles, you have much lower hemoglobin concentration than if you're homozygous for the other genotype. So why did natural selection favor versions of the EPAS1 gene that dampen the acclimatization response? One reason is that we know at all altitudes, women who are pregnant and have very high hemoglobin levels have a higher frequency of miscarriages and a poorer pregnancy outcome. So that would be selection against high hemoglobin. We also know that at altitude in particular, there is a disease called chronic mountain sickness. And chronic mountain sickness is associated with particularly high hemoglobin levels. And Tibetans basically don't get it. They're the ones with the low hemoglobin concentrations, and Andean Highlanders do. So this would be another selective factor against the Andean response that is the same response that we have. Now we can ask whether the same processes occurred in East Africa. And in the interests of time, I will tell you no. That we see the same phenotype of low hemoglobin concentration, but it is accounted for by a different gene. So we have another example of convergent evolution, where two populations came up with independently the same phenotype, the same biological solution, but it was achieved by mutation and selection at different loci.